Hello. Today we'll be discussing human pathology, our third lecture, which is from Robbins and Cotran, Book of Pathology, and we're going to discuss inflammation and repair. When we talk about inflammation, we're talking about the response to infections and damaged tissues, which bring cells and molecules of host defense from circulation to the sites of action to eliminate the offending agents. Components of inflammatory response include blood vessels and leukocytes. The vessels become permeable. Leukocytes traverse the vessel wall. The inflammation may not be mobilized during the active response. The localized effects can become systemic. We talk about acute and chronic inflammation. These are reliant on the microenvironment and the cell composition change. There's afterwards a resolution phase, which consists of repair, remodeling, and removal. Inflammation causes or infections, tissue necrosis, foreign bodies, and immune reaction. Recognition factors are in play, and the host cell has receptors on the membrane, also within endosomes or microbe digestion, and within the cytosol itself. The host cells recognize uric acid, ATP, and potassium leakage as damage indicators, and leukocytes have antibody receptors on their surface. The inflammation mediators include the complement system. There are three components to the acute inflammation phase. First is the vascular response, where there's blood flow increase and increased permeability. Plasma proteins and leukocytes leave circulation and enter tissue. And then finally, there's leukocyte migration and activation at the site of function. Fluid migration, we call this an exudate. This has a high protein concentration and cellular debris. Transudate is, by contrast, an ultrafiltrate of blood plasma. Pus is an exudate, which contains leukocytes. And edema is a fluid excess in cavities or interstitial tissue, and this can either be a transudate or an exudate. So this is a schematic of this process. We have the leukocyte with surface glycoprotein, integrin for a low affinity state. The organism can be noted in here. There are endothelial recognition sites on here, and you can get the recognition of these glycoproteins, the proteoglycans, and the integrin high affinity state kicks in at this point, goes from low affinity to high affinity, and it literally migrates and traverses at the CD31, which is a vascular marker. This goes into the interstitium and travels to the site of the injury where there are microbes here, and cytokines are released, microbes are phagocytized, and you have the macrophage with microbes right here, and cytokines are, these are chemokines rather, and cytokines are released, including TNF, tissue necrosis factor, and interleukin 1. And this process is amplified and repeated. Margination is where the leukocyte attaches orally to the endothelium. It rotates until the receptors are fixed. Rotation is mediated by selectins and regulated by cytokines. Integrins mediate fixation to the endothelium, and these are all regulated by cytokines. Diapedesis is where the leukocyte transmigrates across the vessel. This is also mediated by cytokines. Chemotaxis is a migration to the target site. This is mediated by bacterial products in the microenvironment and endogenous cytokines, complement, and arachidonic acid metabolites. Within 48 hours, monocytes replace neutrophils, and these are longer-lived cells. and there's clearance of the offending agent. So leukocytes are activated, there are increases of calcium, protein kinase C, and phospholipase A. Phagocytosis involves recognition, engulfment, and killing or degradation. The receptors are carbohydrates or glycolipids. These are facilitated by antibiotics. Engulfment occurs when the vesicle is formed from the membrane. This contains the particle in question. It is fused with lysosomes. Following this is intracellular destruction, which is conducted by reactive oxygen intermediates, reactive nitrogen intermediates, and lysosomes, superoxide free radicals. Hydrogen peroxide oxidized to hypochlorite, which is bleach. Nitric oxide is oxidized to free radical peroxide nitrite, which is the formula is given there. Lysosomes contain multiple proteases, which target a variety of exogenous forms. Neutrophil extracellular traps a fibular mesh, which retains antimicrobial substances at the site of action. These are formed by nuclear chromatin derived from leukocytes. Leukocyte-mediated tissue injury is collateral damage. The same mechanism is enforced. This is an unchecked response, and the cytokines amplify or limit the inflammatory response. In some cases, the immune response ends up being worse than the disease in question, depending on the response of the cytokines. Inflammation mediators are a variety of agents. These are secreted by cells or plasma product. They respond to stimuli. They're short-lived, and they can stimulate other mediators. Vasoactive amines includes histamine and serotonin. The former are released from mast cells. They mediate immediate permeability in endothelium. Serotonin has a less well-defined role. Arachidonic metabolites include prostaglandins and leukotrienes. These are released via phospholipase A2, which increases calcium and kinase activity. Prostaglandins are a family of structures. They are vasodilators and inhibitors of platelet aggregation. They facilitate the inflammatory response. Imbalances are implicated in thrombus formation, and they also induce pain and fever. Leukotrienes are chemotactic agents which recruit leukocytes. This is a more painful vasodilator than histamine. Lipoxins inhibit chemotaxis. They're also inhibitors of prostaglandins and leukotrienes. Cyclooxygenase inhibitors include aspirin and non-steroidal anti-inflammatory drugs, NSAIDs. They inhibit COX-1 and COX-2 cyclooxygenases, and COX-2 is more active in inflammation. Some non-steroidal elements or agents target COX-2 preferentially. COX-1 mediates platelets. Corticosteroids act on COX-2, phospholipase A2 cytokines, and nitrogen oxide products. Cytokines and chemokines are a variety of proteins which mediate and regulate the inflammatory response. Tumor necrosis factor and interleukin-1 promote leukocyte adhesion, endothelial activation, leukocyte activation, and the acute phase response, which include fever and sepsis. Chemokines attract leukocytes. These are a family of agents. They're also antagonist medications which exist to counteract these effects. We have the complement system, which is a system of over 20 proteins. A critical step is a cleavage of the C3 protein, which occurs by any of three pathways. The classical pathway is where C1 is affixed to antibody combined with antigen. The alternative pathway is activated by microbial surface molecules without an antibody. And then finally is the lectin pathway, where lectin binds to the microcarbohydrate and activates C1. C3 splits into C3a and C3b. C3b converts C5 to C5a and C5b. C5b binds C5 to C9, and this forms an MAC complex. The A forms, the subset of complements, stimulate histamine release, and C3b also facilitates phagocytosis. This is a cascade reaction, so we're talking about thousands of subsequent reactions from the previous one. Schematic showing this process. We have the alternative pathway here with the microbe attached. C3b is deposited on the microbe. There's also a classical pathway where the antibody mediates this as the microbe here, and the lectin pathway, which is a mannose-binding lectin. In all three cases, they culminate where C3b is deposited on the microbe. You get effector functions, so C5a and C3a create inflammation, where there's recruitment and activation of leukocytes through C3a with destruction of microbes. C3b is phagocytosis, where there's recognition of the bound C3b by the phagocyte C3b receptor, and you get phagocytosis of the microbe. And then finally the MAC complex, where there's a formation of an attack complex, MAC, and this causes lysis of the microbe directly. And again, we have a cascade mechanism tightly regulated. Other inflammatory mediators include platelet activating factor. They are products of coagulation with a tenuous link to inflammation. Kinins include bradykinin, which is an anaphylactic mediator, short-lived. Neuropeptides are involved in pain transmission. Correlations to inflammation. There's an effusion accumulation in body cavities, which is not necessarily inflammatory. A fibrinous exudate has a coagulation meshwork which provides template for fibrosis and blood vessel formation and scars. There is a purulent inflammation and abscess formation. An abscess is a localized collection of pus, and this is characteristic of pyogenic, such as Staph aureus or Streptococcus species bacteria. Ulcers are, by definition, a defect in an epithelial surface with associated inflammation. And the resolution of acute inflammation can be complete resolution with reversion to prior state. With more extensive injury, you can have scarring, and with chronic inflammation, you can have cell composition changes. In chronic inflammation, this is a response of prolonged duration where a combination of phases occurs. The causes are persistent infections, hypersensitivity disorders, and prolonged exposure to toxic agents. Morphologic patterns exist. These are mediated by mononuclear cells, including macrophages, lymphocytes, and plasma cells. There's tissue destruction and healing components, which include blood vessels and fibrosis. Macrophages are the most important cell in chronic inflammation. They secrete cytokines, growth factors, and they activate T lymphocytes. These are derived from monocytes in bone marrow. They are present everywhere. They are localized in certain defined locations, which differentiate into macrophages in tissues. The macrophage phase has a shortened half-life. These are activated by microbial products or cytokines. They induce free radicals and lysosomes. They are also activated by T cell produced interleukins. These are involved in tissue repair and are not part of the inflammatory process. And these effects can create much tissue damage. Lymphocytes predominate in autoimmune and hypersensitivity diseases. They respond to the same array of protein factors as with other cells. CD4 are helper T cells. They secrete cytokines and they amplify inflammatory responses. These are responsible for an array of leukocyte recruitment factors which target different types of microorganisms. The macrophage presents its antigen to T cells, which secrete cytokines, and they create a cascade. Activated B lymphocytes and antibody-producing plasma cells are additionally involved. Eosinophils are seen in allergic reactions and in parasite manifestations. There are granules, which are major basic protein. They target parasites and they create much tissue damage. Mast cells bind IgE antibody, they degranulate, and these release histamine and prostaglandins. And neutrophils can retain their presence in certain disease states as a chronic infiltrate. Another type of inflammation is granulomatous inflammation. This is a specialized type of chronic inflammation characterized by macrophages, T lymphocytes, and occasional necrosis. There are two types. The first of these are foreign body granulomata. These are responses to inert material. You see multinucleated cells and sometimes the foreign material of interest. Then there are immune granulomata, and these occur when antigen is persistent. Cytokines perpetuate the response, and these are seen in certain organism infections such as mycobacteria and fungi, and in some non-infectious diseases such as sarcoidosis and Crohn's disease. And you always exclude infection when there are granulomata identified. These can be done with special stains. Systemic effects of inflammation, you get fever. These are caused by pyrogens. These are effects or manifestations of an acute phase response. Exogenous forms stimulate cytokine release, and they act on prostaglandins to increase fever-setting neurotransmitters. There are also acute phase proteins. These are plasma proteins which increase in acute phase. They include C-reactive proteins, abbreviated CRP, fibrinogen, and amyloid A. CRP fixes complement. Fibrinogen facilitates coagulation, and the erythrocyte sedimentation rate is increased. Leukocytosis, increase of leukocytes has increased production in disease. Neutrophils are seen in bacteria. Lymphocytes increase for viruses, eosinophils in allergies and parasites, and some diseases cause decreases of leukocytes. Other symptoms include increased pulse, blood pressure, rigors, which is shivering, chills, malaise, and somnolence. Ultimately, you can get tissue repair. There's regeneration where the normal state is resumed. You have scar formation where function is retained, but there is replacement to varying degrees in fibrous tissue. The ability to affect tissue repair often depends on the tissue type. So labile tissue is continuously dividing cells such as hematopoietic cells and epithelium. Stable tissues regenerate when necessary. These include solid organs such as the liver, which can get complete regeneration, partial regeneration of kidney and pancreas, and smooth muscle. But permanent tissue cannot regenerate. This includes neural tissue and cardiac muscle. Growth factors originate with macrophages. There's a liver regeneration. This is also mediated by cytokines. They're progenitor cells which contribute to liver regeneration. And the steps in scar formation, there's angiogenesis, where small vessels form larger ones. Granulation tissue is an increase of fibroblasts and neovasculature. And thirdly is remodeling of connective tissue, where granulation tissue is ultimately replaced. This is mediated by macrophages, which remove the dead tissue, and they produce cytokines which stimulate fibroblasts. And the most critical cytokine in this grouping is transforming growth factor beta. Complicating conditions include comorbidities, diabetes, steroids, atherosclerosis, and malnutrition can retard these processes or obviate them completely. Fibrosis is also a pathological factor, which is seen in cirrhotic livers, systemic sclerosis, interstitial fibrosing lung diseases, reactants to toxins in exogenous areas, and kidney. Scar hypertrophy, keloids are a specific form of that and has specific appearance under the microscope. And that's it for the talk. Thank you very much.